I have no disclosures. Okay, so I will quickly start with an introduction, giving the background for this talk. We all know that histocompatibility is essential for transplantation. And we've seen from Professor Naika's talk that a good match minimizes immunological rejection of a graft. Aside from matching the HLA antigens, you also need to ensure that the recipient does not have any donor-specific antibodies, because this also plays a role in graft rejection. Currently, the histocompatibility and immunogenetics testing in Kenya are outsourced to various labs who actually send the specimens to South Africa and India. And we know that as we're expanding our transplant capacity, we also need to increase build capacity and expertise for testing locally so that we can support the solid organ transplant programs, and in the near future, the stem cell transplant programs. So I'll spend the next few slides. Okay, this pointer doesn't seem to be working. Okay, it's back. So this will be the outline of my talk, a brief overview of HLA, and then describe its importance in transplantation. And then I'll talk about the methods of HLA typing and the aim of HLA typing. And then we actually just want to describe to you the validation process we've gone through at KNH, EACI lab, in validating a HLA typing assay, and then we will conclude. So what is HLA? HLA stands for the human leukocyte antigen. These are glycoproteins that are expressed on the surfaces of cells in our body. And the function of HLA is actually to present peptide, broken down antigen, to T lymphocytes. So T lymphocytes are not able to recognize antigen in its native form. It has to be taken up, broken down into smaller pieces, and then displayed to the T lymphocytes so that we can mount a protective immune response. The other role of HLA is in immune regulation. I'm sure we all know that as much as the T lymphocytes need to recognize antigen displayed by HLA, it has to be one's own HLA presenting that antigen. So as the T lymphocytes are developing in the thymus, they're educated to be able to identify our own HLA and then identify our own antigens. So that is the aspect of immune regulation that we call HLA self-restriction. We've already seen the genomic organization of HLA. So these antigens are coded for by a cluster of genes on the short arm of the 6th chromosome. And there are 3 particular classes. We have the classical HLA genes that code for the class 1 and class 2 molecules. So these are the ones that are important in histocompatibility. And then we have the class 3 molecules that are not classical HLA genes. This code for other molecules involved in the immune response, for example, the complement proteins. So a bit more detail on the genes and the gene products. So at the top there, I don't know how to use this pointer. Anyway, so we can see the actual gene as it looks, with the big boxes being the exons. Those are the parts of the gene that code for the specific parts of the proteins. And at the bottom, we have a schematic of how the proteins look when they're expressed on the surface of the cell. And what I want to show here is we can see the peptide binding groove, the small dots. That is the peptide antigen. That is where the antigen sits so that it can be recognized by the T lymphocytes. So for the class 1 HLA, that is coded for by one gene. The one at the top, it's just one gene that codes for the 3 parts of the glycoprotein. And then we have another protein called the beta-2 microglobulin. That is not really, it's just, it's not coded for by the HLA genes. It's just used to stabilize that complex. This protein is, oh, thank you. Okay, so this second exon, third exon, code for the alpha-1 and alpha-2 domains, and that's where the peptide binding group is. The HLA class 1 molecule is expressed on all nucleated cells. For the class 2 genes, we have 2 glycoproteins, an alpha chain and a beta chain, that are coded for by the A gene and the B gene. And it's a second exon that is important for coding for the alpha-1 and alpha and beta-1 domains where the antigen will be displayed. Why I'm highlighting this is because we all know that the HLA genes are very polymorphic. That means there's more than one variant for these genes. So this is where a lot of the polymorphism occurs. And that is important just to ensure that we can bind and present to our T lymphocytes as many different peptide antigens as possible so that we can produce a diverse immune response. The class 2 genes, proteins are expressed on what we call the antigen presenting cells. So B cells, dendritic cells, monocytes, and macrophages, and T cells can also express them when they're activated. So in terms of inheritance of the genes, we inherit them in a Mendelian fashion, and what we inherit is one haplotype from either parent. Okay, and usually this is inherited as a block of all the important genes, the ABC, DPDQ, and DR. It's very rare for there to be a somatic recombination that will cause a change in what the child inherits from the parents. So usually we inherit everything. That is what we call a haplotype. Both the haplotypes inherited from either parent will be expressed. So what is the role of HLA in solid organ transplantation? I've already alluded to the fact that T lymphocytes recognize antigens displayed to them by HLA, but it has to be self-HLA. So any foreign HLA will also be recognized as a foreign component that will mount an immune response. So instead of mounting a helpful response, the response will target that particular HLA. And this HLA can be expressed on the graft because the graft has nucleated cells and it can also have some of these antigen presenting cells. So there are two pathways of recognition. I won't go into details of that. In the early phase, we have the direct recognition where we have what we call the passenger leukocytes, which include some of the antigen presenting cells. They will be on that donor kidney, for example. And if the MHC on those leukocytes is recognized by the recipient's T cells, that is enough. If there's a co-stimulatory signal, that is enough to mount an immune response. In the later phases, because those passenger leukocytes have a lifespan, they could die off. In the later phases, what you have is the HLA from some of these passenger leukocytes, for example, if they're dying off, being taken up by the antigen presenting cells, broken down into peptides and presented by the recipient's antigen presenting cells to the T lymphocytes. And we are able to mount an adaptive immune response, meaning both the humoral response with antibody production and a T cell response with the T helper cells and the cytotoxic T cells being activated. And what we have seen this morning is that the outcomes, clinical outcomes after kidney transplantation are strongly associated with the degree of mismatch between the donor and the recipient. When it comes to solid organ transplantation, the important HLA antigens are the A, B, and the DR, okay? Okay, so aside from the HLA match, we'd also want to ensure that the transplant recipient does not have any antibodies against the donor HLA molecules. And there are various ways in which these antibodies can be produced. These are what we call the sensitization events. Can be from pregnancy. During pregnancy, remember, the fetus will inherit the paternal HLA. So the mother can become sensitized against paternal HLA. We can get them from blood transfusions. These blood products contain white blood cells. Red blood cells really don't express HLA, but the other cells, white blood cells and platelets within the blood product contain the donor's HLA and from prior transplantation. So as I've alluded to, antibodies increase the risk of allograft rejection, and that particular characteristics of these antibodies which increase the risk of rejection, the amount of antibodies, the specificity of the antibodies. So for example, from a blood transfusion, someone can become sensitized, but maybe the antibodies they have do not target the HLA on the actual donor that they're being evaluated with. And then the level of the HLA expression on the graph is also important. And this image is just to highlight the importance of the donor-specific antibodies, antibodies against the HLA on the donor, and the characteristics. For example, if it's a complement binding antibody, that portends even a more severe graft rejection reaction with markedly reduced lifespan. of the graft. The other important aspect is that it's been shown that early recognition of new antibody development, what we call the de novo donor-specific antibodies, when you're monitoring a patient post-transplant, you may be able to start recognizing and probably institute measures to prevent rejection earlier than if you're just waiting for a kidney biopsy to evaluate any rejection responses. Okay, looks like that's a blank slide. So now I'll come to HLA typing, now that we know the basics of HLA. So this is the process of identification of either these molecules or the genotype of HLA within an individual. And usually we perform this for both the donor and the recipient, and then compare the two to look at the degree of match or mismatch. The goal of HLA typing is to provide, when you look at that match or mismatch, you're providing a measure of immunological risk of rejection. And the aim basically is to ensure that the transplant recipient can keep that transplanted organ for the longest amount of time. So there are various typing methods that have evolved over the years with advances in molecular biology. Back in the day, probably in the 1960s, what they performed was serological typing, which is based on antigen-antibody reactions, just like the other serological tests we perform to look at the level of antibodies or antigens in a particular specimen. So they would use panels of serum, which contains antibodies from multiparous women, because these women were exposed to paternal HLA and then come up with different combinations of this serum. That would be the reagent panel. And they would test this on the recipient's B cells and T cells. Remember, the T cells express class 1 HLA, the B cells express both class 1 and 2, but they would use that to compare and see whether someone is sensitized against class 1 or class 2 HLA molecules. And this test is basically called the complement-dependent cytotoxicity test. I'll allude to it later. But because of, it's a very, I don't know if that's a finicky test, very time-consuming test. It's difficult to maintain stability in these panels of antisera. Now they have come up with molecular typing methods, which basically look at the variation at the level of the DNA sequences. So there are three major types of molecular typing methods. There are two that are based on PCR, and then the third type is based on sequencing. So the first PCR-based methods is where they use primers that target specific HLA DNA sequences. So they're already known primers. Remember, we know the human genome and we know the nucleotide sequences in the human genome. So that is where that information came from. And they would use the primers to amplify targeted HLA alleles in the recipient and the donor DNA, and then detect for amplification. This is the system of testing method that is being used at Muhimbili for their solid organ transplant program. For us in Kenya, we want to use the sequence-specific oligonucleotide probes. So the principle is almost the same, it's just the methodology that is slightly different. So we use oligonucleotide probes. Those are short DNA sequences that are labeled with biotin. We amplify the target sequences. So we remember the image I showed you of the gene segments of HLA. So for class 1, we amplify exon 2 and 3. For class 2, we amplify exon 2. And then denature the DNA so that you have single strands and hybridize them to the probes. Okay, and then if there's hybridization, a secondary antibody is added, and then we measure the fluorescent intensity. The third method of typing is a sequence-based typing. These are preferable for bone marrow transplant programs. And there are different methodologies. There's Sanger sequencing, there's next generation sequencing. The West is now moving to what we call third generation sequencing. So this is just to show you what a HLA type looks like depending on the testing methodology. On my right, okay, the right on this side, sorry. What we have is the nomenclature. So usually there'll be the prefix HLA and then the particular gene. So this could be A, B, or the DR, and then a separator and an allele group. So the allele group is just used to identify that particular allele, let me say, depending on the nucleotide sequence that is there. And these are numbered, so it's a number, and this one numbered in order of discovery, okay? And in terms of polymorphism, for the class 1 genes, there are more than 9,000 variants. For the class 2, there are more than 3,000. So the numbers can go on and on. So serological typing and the PCR-based SSO and SSP typing will give your type at this level, at the allele group. High resolution typing or sequence-based typing will give the particular protein. So remember when your basic biochemistry, once you have the nucleotide sequence, you know what amino acids that will translate into, and then the particular protein. So only sequence-based typing can give you this level of resolution. That is what we call high resolution typing. So for serological and the PCR-based ones, we call this low resolution typing. Low resolution typing is enough for solid organ transplantation. So on this side, we see what the typing will look like. So serological will just give you the allele. For the PCR-based typing, it can give different proteins, but it's not enough to differentiate. For example, 2902 from a 2904 or a 2908, okay? So it gives a string of alleles. However, secondary panels can be added to the SSO and SSP typing to enable high resolution. For the sequence-based typing, you know, it's very exact. It gives you the actual protein. Okay, when it comes to the antibody screening and identification, there are various assays that are available. Some are cell-based, meaning we use the donor lymphocytes and test the recipient serum for antibodies against the HLA on the donor lymphocytes. And 2 panels are usually tested, one on B cells and one on T cells. There's one assay where they add complement. If you remember the complement system, once there's an antigen antibody complex that can activate complement, whose major effector function is to cause cell lysis, okay? So if there's antibody against target HLA on the donor cells and you add complement, there'll be lysis, and that can now be evaluated to give a positive test. So that is the complement dependent cytotoxicity assay, can be used for antibody screening, cannot help identify the antibodies really, but it can, this is the basic cross-match that we talk about that is performed pre-transplant. A better, more sensitive assay is the flow cytometry based cross-match, where they still use the donor lymphocytes. And if you get binding of antibodies onto the HLA, we add a secondary antibody that is conjugated to a fluorescence tag, and then we analyze by flow cytometry. So it's more sensitive and less subjective than the cross-match, the CDC cross-match. For the CDC cross-match, one would need to quantify the number of lysed cells and actually just count. So it can be subjective depending on who is doing the counting. And then there are solid phase assays. There are ELISA-based assays, but those are not preferable. What's preferable is these assays where they have purified HLA antigens adsorbed onto polystyrene beads, and then the rest of the testing is the same. So incubate this with the patient sera, the recipient sera, allow for binding, add your fluorescent labeled secondary antibody. So this is anti-human globulin. So it's an antibody against human antibody, so it will bind onto that complex. Okay, I've alluded to the patient-donor cross-match. The principle is similar to what I've mentioned in the antibody screening. Differentiation in terms of what this test is just depends on when it is being done and why it is done. So this is the test, the basically antibody screen that is performed immediately before transplant, and a positive cross-match actually would mean that that donor is the organ from that donor is contraindicated, because this would end up causing hyperacute rejection. So now that we've mentioned the basics of HLA, why HLA is important in transplantation, and the different testing methods, both for HLA and the antibodies, I'm just going to give a brief overview of what we have done to validate this testing at KNH. So validation or verification of tests is an important aspect of implementation of a new lab test for patient testing. So all lab tests, even just a basic UEC panel, has to be verified or validated before we introduce it for patient testing. The terms are used interchangeably, although they're slightly different. For validation, that would be for a completely new test. So for those of us using commercial reagents, we're not developing our own tests in the lab. Usually the manufacturer has already validated the test to give specific performance characteristics. And what you want to ensure is that this test is able to produce reliable results for the intended application. So for the, you can make the right diagnosis using that test. So those are usually done by the manufacturer. So what we tend to do in clinical diagnostic labs is more verification, where we perform the assay as recommended by the manufacturer. So you don't make any modifications. If you want to make any modifications, for example, changing the specimen bottle, you would have to do validation, not verification. So in verification, we just want to confirm that we're doing the test correctly and it performs as indicated. So there are various guidelines for validation and verification. They usually just define the purpose of this process, the scope of the process, but they don't really give a specific approach. So when it comes to the approach, usually the lab professionals would either follow a consensus guideline if there is one. We can look at literature and see what other people have done for that particular, for a similar assay. We can follow the manufacturer's recommendation. That's usually the easiest for verification. But depending on what approach you choose, you have to rationalize the cost. So there are a number of requirements, which I won't go into the details of, but in terms of providing the evidence that you have validated or verified the assay, there are things that have to be analyzed and documented. And I'll just mention what is important for qualitative tests, because HLA typing is a qualitative test. So what we need to document is accuracy, okay, or trueness. So the degree to which the results in your lab are true or conform to like a reference value or a standard sample. So can you detect what the test says you can detect? The other important aspect is precision, which just looks at the degree of similarity between repeated assays. So for example, if you're doing one assay, you can test one specimen 3 times and see how close the results are. Or you can repeat that assay on the same specimen a few days later to see whether you're getting similar results. You can also compare results from 2 different technologies to see the similarity. So reproducibility of the results. So this is the approach that we chose to follow at KNH. We evaluated post-transplant patients who are undergoing follow-up in the renal clinic. Our sample size was 10 patients. We're still in the process of doing this. We haven't really completed it. So we used residual samples. So as they're undergoing follow-up, there's some tests they're usually sent for, and we used residual specimens for this. So we took whole blood in EDTA. That's important because in the validation process, you need to use the actual specimens that you will be using for your clinical testing. And that is what we intend to use. And then the rest of the slide just gives the details of how we went about performing the testing. I won't belabor you with that. So we ran 4 batches. On the first day, we tested 3 patients, and then 1, and then 7, and then 1. And then our approach to analysis was just to look at the concordance of our results to the previous testing that was done. So remember I mentioned that currently we outsource all tests to India or South Africa. For us, most of the patients had been sent to India, but they were tested by the same technique, by SSO. So we just compared our results and the previous test results. We are yet to do the precision assays. So these are our results. So one column just shows what our results of the specific alleles. And remember, this one is one allele and this is the other one. So probably maternal and the paternal. And we compared these results to the other testing labs. We were successful in typing all the samples. So there's no sample that failed. And all our samples were consistent with the previous typing. So there was 100% concordance. So what are we telling you? We basically just want to tell you that at the KNH EACI Transplant Lab, we have successfully verified a HLA typing assay, and we are ready to introduce this test as a clinical service. So you no longer have to think about outsourcing to India or South Africa. Thank you. We have a good turnaround time From the time we receive the sample and we start extraction to analysis of results, it takes 4 hours. So way forward is to complete the precision studies, of course, come up with a rational cost that will also be more affordable for our patients. And then we are going to start the process of verifying the antibody assays. The antibody assays we would like to offer are a pre-transplant antibody screen and identification. If we get a positive screen, we will be offering a cross-match, the CDC cross-match, as well as post-transplant donor-specific antibody monitoring. And I would just like to thank the members of the team. 3 of them are here, as well as these other institutions. Thank you.